Welcome to Life and Biology. I'm Dr. Joel Graff, and this video is going to be about DNA replication. When we talk about DNA replication, one thing to understand is that the strands of DNA have direction to them. You can either talk about going down one strand of DNA in a 5' to 3' direction, or you can talk about going in a 3' to 5' direction. And what I mean by that is if you have two strands of DNA and they're both pointed in the same direction and you've got them running parallel to each other, this is not how DNA looks. They are instead set up so that the two strands that are running parallel to each other are running in opposite directions, so that that structure is called antiparallel. So where do we get this 5' and 3' nomenclature from? And it has to do with what a nucleotide looks like. And so the light's not working with me very well today, but so to understand 5' to 3', we need to learn about counting carbons on the sugar molecule of the nucleotide. What is a nucleotide made of? It's made of a nitrogenous base, so this is either A, C, T, or G. It's made of a ribose sugar. A ribose sugar is a pentose sugar. Pent is short for 5, so this is a 5 carbon sugar. We've got a 1, 2, 3, 4, 5 carbons, and I've got them numbered there, and that is if I ever refer to the 2 carbon, I'm referring to this carbon here. If I'm referring to the 3' carbon, I'm referring to this carbon here. So we could, when the next nucleotide gets added to this nucleotide, its phosphate group is going to come in and attach to that 3' carbon there, and this hydroxyl gets kicked off the molecule. And so then that molecule was just added, that new nucleotide was just added to the 3 carbon of the sugar. And so as we are building the DNA, we move in this, here's the previous nucleotide, here's the current nucleotide, and then the next nucleotide would be here. We're moving in a 5', 4', 3' direction. We can leave 4 out because just saying 5' to 3' makes sense. We don't have to talk about that 4. And then if we were talking about working our way back on a nucleic acid, we would talk about working from a 3 to 4 to 5 carbon direction. This is important because your polymerases all work in a 5' to 3' direction, meaning they always add the next nucleotide onto this 3 carbon here. This 3' hydroxyl is required because you need this oxygen. It's electronegative. You need it pulling away some of the electrons from this 3 carbon so that this 3 carbon has a slight positive charge and can be attacked by that phosphate group of the next nucleotide. Okay, so a little bit of organic chemistry thrown in there for fun. So when we talk about a DNA replication fork, we're talking about starting with one double-stranded DNA and at this end of the figure, and then on this end of the figure, it goes into a replication fork, and you end up with 2 double-stranded DNA out of the deal. And so what happens is you've got a helicase here that's at the leading edge of the replication fork, and it's going to be moving in this direction. Okay, and so it's going to be breaking apart the hydrogen bonds that are formed between the nitrogenous bases in your double-stranded DNA. As it's moving and basically acting like a zipper and unzipping these DNA strands, then we're left with single-stranded DNA, and that gives a chance for a polymerase to come in and make the second strand. And so we've got this strand down here is called the leading strand. This strand up here is called the lagging strand. So let's start with this leading strand first. The brown strand of DNA, the bottom strand of DNA, is running from 3' to 5' in this direction, which means the newly formed nucleic acid is going in a 5' to 3' direction. That means that the polymerase, the DNA-dependent DNA polymerase, is going to be making use of that 3' hydroxyl to add another nucleotide to the 3 carbon of the ribose that had just been laid down on this track. And so it just works, and it basically the helicases going in this direction, and this DNA-dependent DNA polymerase is just following right along behind that helicase. The lagging strand is where things get a little bit interesting, because this top brown strand is 5' to 3' as we go from left to right on the figure, whereas remember the leading strand was 3' to 5'. And so the polymerase here is going to be attaching to the 3' end of this new strand, and if the top strand's going in this direction, the bottom strand needs to be going in this direction for the polymerase to work, which means that as this helicase is going forward, we keep getting new single-stranded DNA, but you don't add DNA, new nucleotides to that 5' end of that strand. And so what happens is I've got drawn in pink here is an RNA primer. An RNA primer gets laid down, and then the RNA, it also has a hydroxyl group at its 3 carbon, and so then it works as what's called a primer, and the polymerase is able to work off of the 3' carbon of that little stretch of RNA, and the polymerase is working in that direction, and it'll go back until it connects to the next piece of DNA. So as this helicase moves in this direction, we're going to have a new gap, and you're going to need a new RNA strand laying down so that you have a 3' hydroxyl that can be added on. And so the lagging strand is a lot more complex, a lot more mental gymnastics to think about than the leading strand. So that was a DNA replication fork, and then I just want to finish up this video with a quick comment on if you have a replication fork and another replication fork and they're set up like this, you get a replication bubble. So here I started with double-stranded brown, that's brown color, hopefully the colors are coming through, double-stranded brown DNA, and it's in a circular shape. Bacteria genomes are often circular shape. The plasmid DNA in a bacteria is often circle shape. So DNA does come in circles as well as straight lines. But up here, I have some of the DNA here on the brown labeled ORI, which stands for origin of replication, and the other's opposite strand is also an origin of replication. So that is where your DNA replication machinery will get recruited when you start to form a copy of DNA. So before any of these orange strands of DNA were there, the 2 brown strands were together and you had your origin of replication. You separate out those 2 strands, and the replication forks work in both directions going around the circle. So I've got an arrow here and an arrow here indicating that those helicases are just going to keep working their way around. This helicase is going to work around in that direction there. And then, so together, they're going to eventually meet up on the opposite side of this circle, and you'll have gone from 1 double-stranded circular piece of DNA to 2 double-stranded circular pieces of DNA. I drew in the helicase, or the DNA-dependent DNA polymerases here in green. I have, this is a leading strand, this is a lagging strand. Over here we've got the leading strand on the bottom and the lagging strand on the top, and so then the polymerase is back up on this left end of the growing chain. So this polymerase will keep going until it connects with this DNA, and at some point it'll drop off. So that is an example of your replication bubble. I think that's about all I want to say for this video. So go ahead and like and subscribe or not. Have a good day.